Good morning, everyone. I welcome you all to this 15th day online CSIR training. Before I enter to the section, please let me know, am I audible? Can you see my slides? Yeah, thank you for the confirmation. Um, Now I am going to enter to the section. Today is the 15th day. It's a second section of our 30-day coaching. In this second section, we are discussing the unit 1, 2, 3 and 7, 8, that concepts. Here in this second section we have 6 units: 1, 2, 3, 6, 11, 8. In that yesterday we finished up the unit 1. Today we are partially 3 by 4th of today we are going to cover the 3 by 4th of the second unit. Okay, the second unit is a cellular organization. In the cellular organizations, first we want to understand the cell outer surface. Okay, cell outer surface, that is cell membrane. Then second step is the part B is a cellular organization. What is the important parts, materials present in the cell inner part? So that might be the cytoplasm, that might be the nucleus. Okay, apart from the understanding of the cell membrane and what is the inner material of cell nucleus cytoplasm, then after the studying, in the main part is a genetic material, that is the gene and chromosome. In the final step, tomorrow we will discuss about the genetic material. These are all the three major parts in unit 2. First is the cell membrane, then cell structure, then genetic material. So they are moving outer to core. Okay, first enter into the cell membrane structure. In all bacteria, human, plant, everywhere there was a cell membrane is there. That cell membrane having the made up of the lipid molecule. Okay, the major component of this material is the fully hydrophobic material. Why it needs hydrophobic material? Hydrophobic layers, hydrophilic layers means that a cell wall need to be stable in the environment. The environment in a cell, in a plant or in an animal, that internal environment is made up of the water. So that cell wall should be insoluble in water. So that made up of the made up of the hydrophobic hydrophilic head and hydrophobic tail. In this bilayer, the internal, that is that bilayer, that hydrophobic tailed and hydrophilic headed molecule is called as the phospholipid bilayer, because that is the lipid molecule having the hydrophobic tail and hydrophilic head. That component made a layer, that layer called as phospholipid bilayer. Then and with that phospholipid bilayer, that having the peripheral membrane protein, integral membrane protein, globular protein, glycoprotein, n number of proteins are there. Then cell membrane receptor proteins. Receptors, all that kind of proteins are there. Why that proteins are needed for needed in the cell membrane? That are the signal channels. Okay, in that cell membrane, that also having the carbohydrate sugar chains and the cholesterol. So only we are telling that that membrane having the polysaccharides. That polysaccharides are the carbohydrate sugar chain. So now for understanding, the cholesterols are present inside the phospholipid bilayer. Carbohydrates are present over the phospholipid bilayer. It's a very important thing. The carbohydrates are present over the lipid bilayer, and cholesterols are present inside the phospholipid bilayer. Here globular protein, glycoproteins are present over the phospholipid bilayer. Integral protein and peripheral membrane proteins are present inside the phospholipid bilayer. It's a basic concept. And cellular receptor proteins present intermediately in the phospholipid bilayer. It's another view of the phospholipid bilayer. For understanding, I am insert this. Okay, what is the role, location? See here I mentioned the location. Okay, that phospholipid bilayer is a main fabric membrane. It's that fabricated membrane. The cholesterol is that tracked between the hydrophobic tail of the membrane phospholipid. Cholesterols it's bound between the hydrophobic tail of the membrane. Integral membrane present embedded with the phospholipid bilayer. That is maybe the both extend. It's not extend to the both side. It's it's present inside, not extended to the bilayers. Peripheral layer is the inner and outer surface of phospholipid bilayers. It's not embedded to the hydrophobic core. Hydrophobic core means that is the outer, that here you can see that hydrophobic means it's internal, so it's not internal, the not touched to that, so it's present over that. Carbohydrate chains, that extracellular side chains. Okay, each and everything it's having a function. This function is defined by various various purpose, various cell cycle activations. This extracellular matrix and cytosolical anchors are it's an own pathway. Then enzyme entry, receptors, why that the receptors have present over the cell membrane. These are all the major need. Okay, extracellular matrix are having the that are cytoskeleton, that is the one outer layer for the cell. The enzymes are helps to for the ATP synthesis and in this complex enzyme target complex formation. Then that enzymes are activating the cell signaling. That receptor means that receptors are cell communication, involved in the cell communication. Then passive transporters are used for the diffusion of the cells, that is maybe ions are then active transporters helps for ion pumps. These are all the major membrane proteins and activation. Okay, this it's are about the imaginary words, right? We are not how we are understand the cell structure. We are that researchers are explained with the models. That model one is sandwich model. That sandwich model called as the Davison and Daniel model. Then fluid mosaic model. These are all the basic structural model. The researchers in the earlier stage they are mentioned they are explain the protein structure, plasma membrane with the models. That models called as that having two types. One is sandwich model, that is another sandwich model is also called as Daniel plus Davison model, Another one is a fluid mosaic model. Why I am this including this means this model based questions are there in repeat nearly 4 or 5 times they are asked in the previous year question, because this in sandwich model they explain about the hydrophilic and the protein structure alone. They explained there is the divalent cations as calciums forms a complex with a lipid or a protein present in the plasma membrane that reduce their interaction with the water. It's a statement of division. So therefore the membrane containing the calciums are less permeable for ions. It's a statement of the sandwich model. But the argument is, do not exclude the possibilities of proteins may be span on the membrane such that the mosaic of protein rich and lipid rich region is formed due to the lack of experimental evidence. They are in that earlier stage, if they don't have that much of facilities with instrumental facilities, they mention their this model with their experiments. Okay, there was some proteins present in the cell membrane and some of the ions present in the membrane. This is a statement of the sandwich model. So important thing is the membrane, the proteins that reduce their interaction with the water. It's a basic keyword in the sandwich model. Then fluid mosaic model. In this fluid mosaic model, that is well organized, well studied structure model, because in this model they explain the integral protein, carbohydrate chains, membranes, integral proteins, all the things. Okay, these are all the basic things of output obtained from the fluid mosaic model. If there the comparison between the sandwich and fluid mosaic, it's well studied, and the integral proteins and surface proteins and carbohydrates, cholesterols are studied in the lipid bilayer, this fluid mosaic model. Then the diffusion through the plasma membrane. Diffusion through the plasma membrane happens in a different way. First one is the lipid soluble molecule entering, then lipid insoluble molecule entering, then lipid insoluble molecule entering through the channels, then water molecule. These are all the 4 different kind of diffusion happen in the cell. Okay, how the lipid soluble molecule enter into the cell? Because that cell wall made up of the lipids, so it's easily attracted by the lipids, so that enter into the membrane, normally enter into the membrane and move to the cytoplasm. But the lipid insoluble molecules are not entered to the lipid bilayer, so that are entered through the facilitated carrier proteins. That carrier proteins are present in between the cell membrane. So it's transporter proteins are receiving the lipid insoluble molecule and release into the cytoplasm. Another model is the channels. These channels are ion based ionic channels. Then water molecules are observed based on the concept osmosis. These are all the 4 concepts: normal diffusion, channels and carriers, then osmosis process. What osmosis process helps to transfer the water molecule. The normal diffusion helps for the lipid exchange. This is another pictorial representation for passive transport. Passive transport means the transfer of the lipid in the outer membrane to inner membrane. Then non-specific transporters, that active transport can be facilitated through the non-specific transporter. These are the normal one kind of protein. These proteins are accepting the molecule, receiving the molecule and release into the cytoplasm. Then facilitated diffusion through the specific receptor. It's a specific receptor for glucose, so that glucose is released by this receptor. This is a transporter substance. This transporter substance are entered into the protein. That proteins are releasing the molecule into the outer surface. These are all the active transport molecules. The process, the lipids are easily diffused through the active transport process. Passive transport process. Then another one is the active transport. It's a facilitated diffusion. In this, it may explain with the pumps, the sodium potassium pumps, because the extracellular space are having the sodium. So internal sodium molecules here that are present inside the cell, due to the attachment of this production of the ATP, that molecules are bound with the ATP bounded molecule is bound with the membrane that automatically close, so the open that makes a open in the outer cell membrane. That outer cell membrane receive the extracellular material after the removal of the production of the ADP, that molecules are enter into the cell. Then next step, the phosphorylation happen. Then that external surface molecule automatically enter into the cytoplasm. It's a basic concept. First the ATP is attached to the membrane. After that the cell the channel open, then receiving of the molecule, then again ADP conversion, thus the release of potassium. Once sodium is moved towards the outer part, so potassium enter towards the inner part, inner cytoplasm. These are all the possibilities of entering and non-entering. Okay, here we need, if there is a gas, means that is easily permeable between the cell membrane. Otherwise the apart from this gases that are not possible to not that gases are not possible to enter, then ethanol can be enter into the permeable membrane. Then water, urea. I want to explain explain the concept here, because the gas is having the that can be enterable into the small portion. How it is ethanol? Ethanol is a mid-polar compound. Polarity of the ethanol is nearly 4.4, that is mid-polar. Polarity is numbered between 0 to 9. 0 means that is non-polar, 9 means that is highly polar. But this ethanol comes and in between that, so that a polarity is mid-polar. So it can be miscible with water and also the lipid. So only the ethanol accepted through the membrane. Then water and urea. Here you can understand, if there is a higher amount of water into the cell membrane, that automatically blast. So that amount entry is a restricted one, that's so slightly permeable. Then glucose and fructose are not directly enter into the lipid layer. So we need the intermediate transporter, that is the glucose transporter molecule. Then ions, cell can permeate potassium, magnesium, calcium, chlorine, and carbonate and phosphate. Here this all ions are enter through the ionic channels. The charge spaced molecules are also in the permeable in the cell membrane. The charged polar molecule, so the lipid is the non-polar, but charged polar molecule, how it enter into the cell, that can be entered through the channels. That channels are pumps that helps that are receiving the amino acid and release on the into the cell. It can be allow the proteins, it can be allow the nucleic acids that are happen through the cell cell membrane transporters. Then the important concept here is the osmosis process. First is the normal diffusion. that normal diffusion helps to transfer the lipid. This osmosis process helps to enter the water into cell. This water transfer is happen in three ways. This hypotonic, isotonic and hypertonic. Hypotonic means that water concentration is higher in the it's higher in the inside, so that want to share the water to external surface. The isotonic means both sides are equal in nature. Hypertonic is a higher quantity present in the outer membrane. These are all the three concepts. How it is transferred? Once the osmosis happen in the cell, it's a basic osmosis, a movement of water across the cell membrane. This is also the transport of by the diffusion. This diffusion happen with the membrane transporter. Here the partially negative charged head and partially positive charged tail are present in the water molecule. This water molecules water molecule present in the in and out of the cell unit. This solid concentration is want to be balanced, otherwise it may be the blast or stringent. It's very thin. The balance of water molecule present in the outside and the inside of the cell want to be maintained in a constant level, otherwise that may be stringent in nature, or that may be the absorbed more molecule, and if inner having the higher quantity of water molecule, that may be the make a blast. So that is a very important thing here. So for balancing, the water molecules are automatically accept and remove from the cytoplasm. It's attached on the protein, that cell surface transporter allowing the water and make a balance. Okay, first then the important thing that we discussed two things, that is the first we having a 4 in the transport diffusion, having 4 different lipid, water and pumps and channels. Okay, this lipids are normally transport, waters are transferred by the osmosis. Now we are going to enter into the pumps and channels. This layers having the intrinsically impermeable to ions and polar molecule. These membranes are intrinsically impermeable to ions and polar molecules, right? Because that cell membrane is made up of the non-polar heads, so that are impermeable to membrane. So we are having two types of membrane proteins, that are all the pumps and channels. Pumps as a energy source. Okay, use the energy source to activation. That pump is a protein, that is one of kind of protein that is called as pump. That pump is it's utilizing the source of ATP or light. It's driving the thermodynamic energy for transporting the ions and molecule. So please understand and remember it. Pumps are using the source light and ATP. These are activated by the thermodynamic law and transport the ion and molecules. Then channels. These channels are channels are downhill or passive transport facilitated relation. These channels are downhill means it can allow and it can exit, allow the entry and exit. That are the ion channels. So first you have discussed, right? Sodium can move towards outer part, the potassium enter to the outer part. The pumps are allowing the only uphill, channels allowing the downhill. These are all the ion channels. These proteins may be the potassium ion allowing protein, sodium allowing protein, are calcium allowing protein. These channels are having the extracellular having the protein that may be attached on in between the membrane and is extended to the outer surface. This may be the happen in there are different type of channels are there in ion channel type. In this ion channel having the 5 subunits in the structure: alpha, beta, gamma, delta and kappa. Then it can be openable. It can this are it's an open form, it's a closed form. Why? How it is open and closed? That having the messengers and receptors. Okay, if the messengers receiving the signals, okay, we need a demand, we need to open that. If that open for the allowing the ion molecule from cytoplasm to external means that is the that is the signal membrane signal. That membrane signal are received at the outer surface and released in the inner surface. That opening that helps for the opening and closing. Here we having 4 types of ion channels. In that all ion channels that having the 4 type. Okay, why need it 4 types? Various cells having various need. So we, because depends on the various need, we having different type of the ion channels. Okay, these ion channels are resting potassium channel, voltagegated channel, ligand-gated channel and signal-gated channels. Okay, why we need it in different form means some of them it may be the directly allowing only the potassium. That potassium it's only the process is always open, that may be enter and exit, that one kind. Another one is the voltage gated channel. This voltage gated channels are responsible for rapid influx of sodium ions during the action of potential that in the membrane, nerves, muscles and endocrine cells. So it's help for rapid influx of sodium. Why the sodium ions are helpful for always helpful in a nature. This is the channel. This channel is allowing the sodium ions while for it's closed at the resting potential. The channel is closed, that open for nerve impulse, the gate open and sodium ions are entered. Then another next step after entering of the particular amount of sodium, that automatically closed. Then ligand gated chain. This ligand gated chains are having the external message receiver receptors. Once it receiving the receptor means that having the signal for the entry. Okay, there is a demand in the cell surface, so we cell in the cell internal cell, so we need to open and allow the ions, so that signals are passed, that ligands are bound with the external surface protein in the channel. That channel automatically open and allow the ions. After the particular amount of ions received into the cytoplasm, that ligand is automatically removed from the receptor, so that are stopped that are automatically closed. These are all the ligand gated channels. Then voltage gated. This is the first thing. I am put the another image. So voltage based thing. There was this external surface around the molecule is if there is a positive charge, because that positive charged molecule is it's bound with, it's having the signaling. The inner it's this vestibules. If we want to the molecule is here, if you want to allow the exit means you want to open this area, right? For opening, that need the charge, negative charge, because negative charge and positive charge only interact with each other. So there was some the change in the electric pulse. So there was negative charge is there, that positive charge and negative charge are attract each other, and it's allowing the release of molecule. After releasing that there was no that relative region, so the positive charge automatically come down and interact, observing the that closing part. Okay, these are all the main thing in the channels. In the in the channels, then we having the pumps. In the cell membrane pumps, we are having the active and helps for the both active and passive transport. These membranes are very it's called as active. It's assist the active transport for referred cell membrane pumps. This cell membrane pumps are activated, allowing the sodium, allowing the potassium, and also ions. Here that sodium potassium pump is there. This sodium potassium pump, once attachment of ADP, that releasing the Na plus ions. After removal of the phosphate group, so that converted to the ADP, that ADP formation helps to allow the potassium ions. The step by step process of sodium potassium pump, it's similar to the voltagegated that's previously I have discussed in the before. Here its process, yeah, it's a similar process. This active transport, that active transport happen with the pumps. This pump is having 4 different steps. First, there is a need of sodium, so the sodiums are moved to the protein and stored. Then ATP comes and bound with the protein, membrane protein. That ATP undergone the phosphorylation. This phosphorylation process helps to produce the ADP. So this phosphorylated proteins make the open in the outer surface. So Na plus ions are removed. Then due to the opening, the potassium ions are enter into the cell. After the entering of the potassium, that phosphate groups are removed from this protein, so that makes a open. That open allowing the potassium. Okay, now we are studied cell membrane, about the cell membrane proteins and cell membrane channels. Now we are going to enter into the topic, the protein sorting. How it is decided? These are the molecule wanted, these are all the molecules are non-wanted things. This is all about the protein sorting. So allowing the mechanism, the sorting mechanism by which cell transport proteins to appropriate position in this cell or outside of it. The sorting target can be inner space of an organelle. What is the need of organ? That based on that only that protein sorting happens in the cell. Okay, this protein sorting, in the protein sorting, if there is a need in the cell, means that is to make a unfold, that the fold and unfolds. Here if the cell unfolded cells having the only one signal region, that signal region it's present at the amino acid end, that during the folding that cause the signal to one particular region alone. That is one kind of cell sorting. Another one is a region having the different signal patches. That signal patches are inactive, that is not receiving the signal while they are in the separate position. Once that all the patches come together and folded in one portion, that signals are passing the perfect signals to all for the process. So this is a kind of cell sorting happen in the cell. The cell, all the intracellular proteins. Here you can see that protein in the nucleus and also mitochondria. The protein helps for the endoplasmic reticulum. That endoplasmic these are all the process. Once one normal protein present into the cytoplasm, that once enter into the mitochondrium, that play a various role, and it's enter into the nucleus means that may be the various role. After that, that folded protein or one new formed protein is made up of the endoplasmic reticulum. That then after the entry of protein in the endoplasmic reticulum, that form as a new protein. That new protein and says one important compound for the Golgi complex. That Golgi complex utilizing the protein for various purpose. That proteins that Golgi apparatus producing the various packaged proteins. It's a sequential process, cell sorting, based on the need of various part of cell. That proteins are getting folded, unfolded form, and various formations are happen in this cell. It's one basic thing. What is the intracell, that the final cell surface proteins are helpful for the R entry. These are all the electrical properties of next here first I wish to tell this because it's a very important thing. Actually in the first image you can see the active passive transport. You can allow and release of Na and K pumps. In this the using of this pump, it keeps the inside cells, the sodium inside the cells is low and potassium is high. Inside of the cell we need to maintain low sodium and high potassium. Okay, in that way that needs some amount with this extracellular surface molecule concentration and intracellular molecule concentration. It's a very important thing. Please note it down because this value, the psi value, sodium ion, potassium ion, pH, carbonate concentration, and chlorine concentration, calcium concentration. These are all very important thing in the cell surface and cell interior and outer part. The outer part concentrations are here. The sodium is having in the higher concentration in external surface, that is 145 millimolar, but inside of the cell that having the 15 millimolar. Potassium, the voltage of the millivolt electricity, the psi value is the 0 in the external surface, but internal surface that having the minus 60 millivolt. Then potassium concentration in the outer cell surface is a 4.5 millimolar, but inner that potassium concentration is of 120 millimolar. pH of the surface extracellular is 7.4, inside the cell 7.2. This carbonate concentration is 24 millimolar. Here inner cell having the 15 millimolar. Chlorine having 116 millimolar. Here 20 millimolar. And then calcium is a 10 power minus 3, and that inside the cell 10 power minus 7 millimolar. These are all the very important thing in the cell surface. That we all by the bicarbonate concentration also very important in the cell signaling, because the concentration want to be maintained maintained based on the glucose transport. The bicarbonates are or balanced by the glucose transporters, and the chlorine concentration is maintained by the that receptors responsible, and calcium sodium channel and potassium sodium channels are very important. These are all the things playing a role. The organelle calcium hydrogen pump, it's present in the nucleus. That also very important thing. That nucleus pump is releasing of hydrogen to that nucleus to cytoplasm, and allowing the entry of calcium from external surface, that cytoplasm into the nucleus. This having the electrical property of membrane. Electrical property of membrane is it's due to the gradient of that potassium and sodium channels. So maintained by the potassium leak channel and sodium potassium ATPase. It's this electrical property is maintained by potassium leak channel and sodium potassium ATPase. Here the voltage gated channels are always in the closed form, that is in the normal resting channel, resting time, that is always in the closed form. If there is a demand, if that receiving the impulse, nerve impulse, then only that will open and release the sodium from cytoplasm to the external surface, and allowing the potassium ions into the cells. Okay, here we having the action potential and membrane potential. These two concept is very important in the very important in the cell membrane. Here the active potential is initiated by the stimulus and generated by the sodium potassium voltage gated channels. Here that polarization and repolarization, that repolarization happens these two processes are normally happen in the channels: polarization and repolarization and depolarization. Depolarization means that is allowing the molecule, and there is no that there is no polarization in the channel. Then repolarization, after releasing of the molecule, that automatically obtaining the polarity, that helps to allowing the ions. These are all the major things happen in the active potentials. Then membrane potential. This membrane potential is difference in the electrical charge between the extracellular matrix and cytoplasm, because that is the charge. The external surface is having the positive charge, the internal surface having the negative charge. Once you are receiving the signals from external surface and outer surface, that makes some charge imbalance. That imbalance is overcome and arranged by the balanced by the ion channels. Okay, here there is a high it's a minus are there, right? If there is changes in the ions, that makes imbalance. If here you having the minus ions, means plus ions, means there is some imbalance, so the pump is automatically get alert. So we need the signal, we need the molecule, then it's automatically open and allow the external molecule. Next, till now we have discussed the outer cell surface of the protein. It's a overall thing. Now we are entering into the internal cell surface. Internal cell surface having the different part. We all know. I'm not just going to explain this images, because we all know the cell membrane, chromatin, nucleus, nucleus and nuclei. Nucleus is a internal chromosome linked molecule. That nuclei is a region nuclei. It's an internal part. Then smooth endoplasmic reticulum, Golgi apparatus, Golgi vesicles, cytoplasm, lysosome, microtubules, centrosome, vacuoles, ribosomes, rough endoplasmic reticulum and smooth endoplasmic reticulum. That rough endoplasmic reticulum, it's immediate to the nucleus. The smooth endoplasmic reticulum, it's over the rough endoplasmic reticulum. Then mitochondria. So these are all the major components. Compared to this, that plant cells are having the external cell surface, extracellular layer, cell wall. Okay, these are all the organs and their functions. It's a very important thing. Cell membrane, it's a outer cell line, it's for protection. Then cytoplasm, it's a jelly-like lipid. This organs, it's a floating of cellular organs that need the cytoplasm. The nucleus, control center, nuclear membrane, it's enclosed protecting nucleus is protects the ribosome. Nucleus is produce the ribosome, right? That is production of ribosome itself for the organs. That then the DNA, it's a heritage material. Chromosome, it's a thread-like structure. It's the of DNA. Normal DNA, it forms the chromosomes. Then vacuoles, that stores nutrition and waste. Mitochondria, it's a jelly bean shaped that produce the energy. Mitochondria is an energy source of cell that produce the ATP energy. Then ribosome, that where proteins are made. Then endoplasmic reticulum, it's called as smooth and soft. That rough and smooth. That rough has the ribosome, smooth don't have a ribosome. Then Golgi apparatus, it's a packing center that modifying the protein and producing the various purpose, producing various proteins for various purpose. Then microtubules, it's for moving, that protein filament involved in the cell movement and shape. Then centrioles, it's contained in the microtubules involved in the cell division. Lysosome, it's responsible for intracellular digestion. These are all the very important organelle parts and their function. Okay, next is major things in the animal and plant: nucleus, nuclei, nuclear membrane and nuclear pores. What is the function of cell wall? It is helpful prevent the enlargement, absorption and transport of the molecule, and it helps to define shape and size, provide the protection for physical injuries to organelles, and it is together with vacuole prevent the skeletal support. Then after the cell wall function and cell wall understanding, we want to know about the nucleus. Nucleus is a very very important region present in the cell. It's located in the center. It helps to the eukaryotes are having the nucleus, prokaryotes don't having the nucleus. It contains all the genetic material in the DNA. This DNA present inside the nucleus. Eukaryotes having, prokaryotes don't having. Then mitochondria. This mitochondria is playing a important role. It's cellular respiration process. It's generating ATP, breakdown of sugars and fats and other fuels. It's obtaining the ATP by the breakdown of sugars, fatty acids and fuels. In the presence of oxygen, it's producing this all the ATP generation process. This breakdown of the large molecule to small molecule is called as the catabolism. If there is a metabolism, the metabolism is it's involved in the mitochondria. In this specifically, that catabolism happens in the mitochondria. It generate energy in the presence of oxygen. That is called as the aerobic respiration. If there is in some cells there was some oxygen is generated that energy is generated in the absence of oxygen, that is called as anaerobic. Here the mitochondria, it generates the energy presence of oxygen, so it's aerobic respiration. These are all the keywords about the mitochondria. It's in this mitochondria, this is a mitochondrial structure. In this mitochondrial structure we can see different regions. This mitochondrial gene express the translation and translations process. This is an overall function. It helps for synthesis of FES cluster and cofactor. This mitochondrial membrane, the metabolism of amino acids or lipids happen in these mitochondria. It's mitochondria having the transport of ions, metabolites, energy metabolism pathway, TCA cycle happen in the mitochondria. It's having the respiratory chains. It having the protein transport processing happen in the mitochondria. Then membrane architecture and dynamics depends on the mitochondrial signaling. This redox signaling and redox process happen in the mitochondria. Main thing is apoptosis signal is transport through the mitochondria. These are all the major functions of mitochondria. It produces ATP. Its regulation of innate immunity. Calcium hemostasis happens due to the mitochondria. Then programmed cell death, that is apoptosis signal is happen by the mitochondria. Stem cell regulation also happen by the mitochondria. So it's playing a vital role. Production of ATP, regulation of innate immunity, calcium hemostasis, and programmed cell death and stem cell regulation. Then the another part is Golgi apparatus. This Golgi apparatus having the it helps to modify the protein. It helps to produce the protein sorting. The last step of protein sorting we discussed about Golgi apparatus, right? This Golgi apparatus having different new vesicles formed in the cell. The formation helps for the new vesicle formation. You can see here how it is formed. The modified proteins and lipids are happen in the Golgi apparatus. The process materials are removed by the cell, it making the mucus and its packaging material. Function is producing huge amount of macromolecules. It's modifying the macromolecule, that is a protein. It's synthesized by the cell, and secretion process that within the cell happen. It's in the cell. This are all about the this Golgi apparatus. It's also produce the lysosome. Okay, what output of the Golgi apparatus is one thing is a lysosome. This lysosome this contains enzymes that break down and recycle the cell components. Lyse means that is the degrade, breakdown. The cell breakdown and the cell lysosome is exploded. This found in the plant and animal cells also. This lysosomes, it's present in the cytoplasm. The function is digest the protein, food, water, and warm out cell parts, the unwanted things in the cells. It's mainly found in the major role playing in the animal. The formation of lysosome, here how it is formed the Golgi apparatus, that having the primary lysosome after the particular period automatically detached from the cytoplasm. Golgi apparatus, it's a basic thing. It's a rough surface endoplasmic reticulum. From that is the cytoplasm Golgi apparatus is formed. Then that removal of lysosome. This endoplasmic reticulum. Now it's endoplasmic reticulum is another key role playing part in the cell. In this function we having the complement proteins. After the help of complete the proteins after the ribosome built them. It's help how it is help them means it makes some membrane. The endoplasmic it's a reticulum is a membrane to the nucleus. It's over to the nucleus. It makes a membrane. This ribosome it attached to and works on the protein. That is called as rough surface. It's a make a membrane alone means that is a smooth surface. Here it's a endoplasmic reticulum. It's a three-dimensional view and it's a how it is arranged. This external surface, this are all called as the smooth. The internal, the near to the nucleus membrane, attached to the nucleus membrane, that is called as the rough endoplasmic reticulum. It's this rough endoplasmic reticulum having the ribosomes. This smooth don't have the ribosome. It's a lack. Okay, this makes the protein used in the cell. It makes the smooth endoplasmic reticulum that makes protein used in the cell. But this for this rough endoplasmic reticulum that export the protein, export to the proteins to export. This function of smooth endoplasmic reticulum is the helpful for lipid metabolism, carbohydrate metabolism, or detoxification, especially for the abundance in the mammalian liver. Where it is helpful one means mammalian liver and gonad cells. It's also synthesis the phospholipids for the cell surface, cell membrane designing. What is the function of the rough endoplasmic reticulum? This the function of it's helpful for protein secretion cell. It's packaged into the transport vesicles. These are all the very important thing. In the rough endoplasmic reticulum, protein packaged into the transport vesicles. Here you can see here transport vesicles, secreted vesicles. This the transport vesicles are attached to the particular region of endoplasmic reticulum. Here the secreted attached to the Golgi apparatus. You can see here that Golgi apparatus it forms and release to the plasma membrane. After this is the sequential process. Okay, another important thing is a peroxisome, the organ. The particular organ that they are particles of about 100 to 500 nanometer diameter. This lipid bilayer surrounded which regulates the enter of and exit of peroxisome. Because this is also made up of the lipid bilayer, so it can be easily enter and release. Then inside a dense matrix, inside part having the dense matrix, that urate oxidase crystalline core. That internal peroxisome structure is called as urate oxidase crystalline core. This internal core have made up of within the 32 peroxine proteins. This internal core that is made up with the 32 peroxine protein. There is a peroxisomal protein that exclude the peroxisomal function inside the organs. What is the function? This biosynthesis of plasmogen, either the phospholipids which the necessary of the normal function of brain and lungs. Whereas this peroxisome playing a major role means that is helpful for plasmogen synthesis. It's also produced that's helpful for the synthesis of unsaturated fatty acids. This having different functions in the different region. Here the hydrogen peroxide is poison, but the peroxisomes having convert the hydrogen peroxides into the water. Then that another function in the breaking of fatty acid into the small molecule in the mitochondria for utilization, for the mitochondrial utilization. Mitochondria needed the ATP, right? The sources we that need the mite source. So here that are the breakdown of obtained by the breakdown of the fatty acid. Another one is detoxification of alcohol, then immediate production of the phospholipid, which typically used to form the membrane. The four different things are the applications are here: plasmogen production, fatty acid synthesis, unsaturated fatty acid synthesis, fatty acid breakdown, hydrogen peroxide conversion into water, then alcohol detoxifying alcohol, then the production of the phospholipid. Okay, the lysosomes also having the major role. What is the difference between the lysosome and peroxisome? This lysosome is filled with enzymes and breakdown and digest organs, but peroxisome is helpful for detoxification of hydrogen peroxide. It's a oxidative breakdown of fatty acid in a normal cell. It's doing the oxidative breakdown. In the lysosome structure, that internal part having the hydrophilic enzyme mixture, but the in the peroxisome, that inner part only having the urated oxidase crystalline core. The structure of the lysosome and peroxisome, it's a little bit similar, but the internal surface of lysosome having the hydrolytic enzyme mixture, but the peroxisome internal structure having the urate oxidase crystalline core. Then that plastids are another important part present in the plant cells that found in plant. The function is store the pigment and food. Plastid, its a functional role is storage of pigment and color food. The structure having hollow sacs around that structure, it's a double membrane structure containing the own DNA, that each and everything having the own DNA. Here what is that this having the three different, the leucoplast, it's helpful for storage of foods, chloroplast, it's helpful for photosynthesis, chromoplast, it's helpful for storage of color. These are all the three different plastids. The plastids are leucoplast, chloroplast and chromoplast. Leucoplast is helpful for storage of food, chloroplast is helpful for photosynthesis, chromoplast is helpful for the color pigmentations storage. These are all the very different role playing. Here idioplast, this is the crystalline, it's a precursor for chlorophyll. Crystalline prolamellar bodies, this helpful for precursor of chlorophylls. This chromoplast is a pigment synthesis and storage. Chloroplast is a photosynthesis and lipid synthesis helpful region. The leucoplast synthesis the fatty acids and amino acids, that are storage. That amyloplast is the storage of starch and sugar. Elaioplast is the storage of lipids and oils. Proteinoplast, it's a site of enzyme activity. Here these functions are very very important. The CSIR question in every year they asked, based on this topic they asked one question. It's very very important thing. Idioplast, it's a cellular crystalline crystalline structure prolamellar body, it's a precursor for the chlorophyll synthesis. It's then chromoplast, it's a pigment synthesis and storage. Chloroplast is a photosynthesis and lipid synthesis. Leucoplast, it's a fatty acid and amino acid synthesis. Amyloplast, it's helpful for storage and sugar. Elaioplast is a storage of lipids, and proteinoplast, it's a site of proteins activity. Then another thing is the vacuoles. Why there is the vacuole presence in there? These vacuoles are stored in water and function is to store the water, salt, proteins and carbohydrates inside a cell. It's having sac-like structure. Plant cells have one large central vacuole. but animal cells having many small vacuoles. These are all the vacuole cells. It's a very large in plant, that's a small size in the animal. It's useful for storage. Then chloroplast structure is a very important thing, because this having the vital role in the cell that lamella act like skeleton of the chloroplast. In the it's a you can find the region, it's a connectors distance between the thylakoids. It's maintain the lamellae. It's helpful for maintaining the distance between the thylakoids which present in the chloroplast. Stroma, it's a space outside the thylakoid membrane. Okay, it's location of the Kelvin cycle. The Kelvin cycle happen inside the chloroplast. It's organic compound forming, helpful for the organic product forming. The granome is stalk of thylakoids, it's grana. Then photosystem, it's cluster pigments and proteins that are absorb the light and are found in the sac-like photosynthesis membrane, that is called as thylakoid. It's a sac-like membrane that is called as the thylakoid. It's helpful for the photosystems, clusters and clusters of pigments, for every purpose. It's having the having the ability to absorb the light, that is the thylakoid. Then the chloroplast, they are having the four different region, that is the lamellae, the stroma, that granum and thylakoid. This are again the explanation about the chloroplast, because it's a image, it's well defined. The stroma, it's empty place. The inner plates of inner membrane, outer membrane, granum is in present, and lumen is the internal part of the thylakoid. It's also the arrangement of each and every is called as the thylakoid. It's arranged in order. Then as cytoskeletal diagram. The cytoskeletal diagram of means it's actually what is the structure of the cell, that each and every cells having the internal cytoskeletons. It's having a plasma membrane, endoplasmic reticulum, it's connected with the intermediate filaments and ribosomes, mitochondria and microtubules and filament. Here our cytoskeletals major thing is the intermediate filaments, microtubules and microfilaments. What is the role of these all things? This cytoskeletal structure is a network of thin and fiber element. It's made up of the microtubules, filaments and fibers. This active function support the system of organs. All the systems want to be in a float format, right? That system is maintained that need a support. That support helpful for the cytoskeleton helpful for the support. Then it helps for maintaining the shape. This specifically it having the three different function, also active filaments, microtubules and intermediate filaments. The actin filaments are twisted one, two strand. It's in 8 to 9 nanometer diameter. It's a very important thing, please listen. Then it's a hollow tube. Then microtubules are a hollow tube. It having the this blue color one is a microtubule that's having the 24 nanometer diameter. Then intermediate filaments are rope like structure. It's having the nearly 10 nanometer. Each and every function, this composed of protein, actin, protein subunit responsible for cellular contraction, crawling and pinching. The actin filaments is made up of the actin proteins. It's helpful for contraction, crawling and pinching. Then microtubules provide the organizations to the cell and move material within this cell. Okay, that want the movement of the ions, proteins in between the organs, right? So that need the tubulin protein subunit. That macrotubules is made up of the tubulin protein subunits. That intermediate filaments provide the structural stability. Uh, that proteins are mimetin subunits. These are all the functions and the structure. That main function of the microtubules and microfilaments are motility. That intermediate filaments help for anchorage. That anchorage means that that structure formation, stability. Okay, till now we discussed about the cellular organs and what is the main portion of cell membrane. Tomorrow we will discuss about what is the genetic information. Here you can this all the organization of the gene, cell division, cell cycle. These are all the main thing. So it takes some time to discuss, and we will discuss about the RNA replication, the DNA synthesis process. That is the replication process and translation process, a transcription process. We will discuss tomorrow class. Thank you everyone. If you have any queries, please post in chat box. Yeah, why the Harinathan question is why only one value is negative? Because the charge is calculated based on the ions. That ions carries, if that chlorine means that carries the positive ions, negative ions. That is, if there is a sodium means that carries the positive ions, right? Based on the positive ions and channel ion is ionic interaction based system helps to calculate the charge. That's why only they mention minus 60 millivolt. Kindly explain the potassium leak channel, ma'am. The potassium leak channel is nothing but that is the sodium potassium channel, because the potassium leak channels are activated based on the sodium release. Once the sodium get released from the internal surface to outer, the cytoplasm to extracellular means that helps to allowing the sodium into the internal portion. Calcium chemostasis is nothing but if the calcium it's want to maintain calcium and phosphorus need to be maintained in the blood. If there is imbalance happen in the calcium calcium imbalance happens in the blood, that blood stream regulate that which regulates the calcium chemostasis. If there is a correct imbalanced means that is the healthy skeletal system. If there is an imbalance in the bloodstream, that makes the calcium chemostasis. It's depends on the it's related to the calcium intake of the channel. That cells need to be maintain the calcium. If there is a higher amount of calcium inside the cell means that makes some hemostasis process. That it's related to the oral intake of calcium, and also the that the calcium is always related to the phosphate balance. Calcium and phosphate want to be maintained in the order, otherwise it makes some hemostasis process. catalase enzyme catalase present in the human cells that convert the hydrogen peroxide into water. Catalase present in the human cells that cell organs that provide that converting hydrogen peroxide into water. Okay, thank you everyone. Thank you for your patience. Tomorrow we will see the remaining portion of unit 2 and unit 3.